Thank you very much, Nancy. Hello, everybody. My name is Yasunori Aizawa. Sorry, it's coming. Could you switch? Yeah, thank you very much. So again, my name is Yasunori Aizawa. Please call me just simply Yas. Yeah, I came from the other side of the North Hemisphere. So it's kind of bedtime for me right now, but please focus on what I'm telling right now. So I think I'm still don't understand what pilot project means. I was selected as one of the suggestion people about pilot project. But my interpretation is that we are just showing our ideas, which will be very important for the future of the Human Genome Project. And then maybe I'm correcting the people who are interested in this kind of theme. And then maybe we are making some small umbrella under the big umbrella of the GP Right Consortium to push the first phase of this GP Right project. That's my understanding, probably correct, right? Okay. So my title is that Scientific Screening for Essential Introns and Retroelements in Human and Animal Cells. So actually as a first presenter of a pilot project, focusing on the human or other big genome synthesis project, I think my role is to remind you that the difference between the genomes that have been synthesized and the ones that are going to be synthesized. So here I show that is there any pointers here? But anyway, so top panel and the bottom panel. So you saw that the genome browser, which has the same length, 300 kilobases there. And it's obviously on the top panel, you see the many genes in the east. There is roughly 160 genes there. So it's kind of a forest of genes there. On the other hand, the top panel, that this is the genome we are going to synthesize, is roughly there are only 4 genes there. And then the bottom part here, instead of having many genes there, we have many retrotransposon sequences there. It's kind of it's derived from the virus genes that jumped in the human genome during the long evolutionary time. So I think the genome we are synthesizing right now is like this. And also, if you look at the gene structure there in human, they are made long introns inside this. But most of the genes in the east doesn't have introns. So that's the difference also. That's a big part of the difference between the yeast genome and the human genome. So that, as you can see, it's a kind of illustration always to show the difference of the ecosystem of the genome between the genome which has many genes and then which the ones has few genes. So that the questions actually we have right now is to see and understand the ecosystem and then the design principle of how the genome under below are constructed. And then the approach I'm taking right now is very simple. Basically, we're making some mutant fragment which doesn't have introns there, and then we replace the fragment into the wild type in a living chromosome. And in this case, we are removing some introns which exist commonly in all splice isoforms. And also, another mutant we are making is transposon-free fragment. So that way, in the beginning, we can test these removal will impact on some expression of the genes, target genes. In this case, you show that the P53 genes. And also, we want to see the expression of at RNA level, and also protein level, and also epigenetic status, and to see the effect of these junk DNA there. And also, we have to use some marker to select out the genes which has a replacement. So we have made this kind of marker below here. So actually, Harvard group, including Dr. Church, already published this kind of marker, which use the piggyback again, DNA transposon sequence, piggyback terminal repeat. So once we have that marker in the living cells, by overexpressing some of the piggyback enzyme there, you can eliminate the whole marker without any scar there. So basically, we can just swap the wild-type DNA part with the synthetic ones. And then the genes we are targeting first is the cancer-related gene in a human. There are roughly 250 genes registered in some cancer gene database. But in the beginning, we are focusing on these 5 genes, which are different in the length of the gene, like this. And then, as Jeff mentioned, one of the purposes for the first phase of the GPRISE project is technological development. So that, I think we are making some progress and then solve the problem on some technical issues that we cannot see right now. And then once we develop the technique, maybe we want to apply the basic strategy to the different animal system. Okay. And this is the last slide. So of course, by doing this project, we want to see, we are going to see some principle how retroelements and introns sustain the ecosystem of the gene expression. But also, this is important for everybody in this room. For example, we can sophisticate our genome design by learning patterns of disposable introns and retroelements. And also second, this is very important. By removing the repeats, we can reduce the risk and the labor at amplicon assembly and the sequencing check, which are troublesome for GPRISE reader, right? And also, in the end, for example, if we can remove the most part of retroelements, we can downsizing the human genome, the animal genomes, which save money, time, and labor. With that, I want to take questions. Thank you very much for your attention. Any questions?
On which basis do you believe that the introns and retroelements are dispensable in a live cell?
Well, my guess is that probably some research in Drosophila showed that some introns has, enhancer sequences for transcription. So that probably some of the introns are important for the expression regulation. And also, we know we have many reports on some important retroelements for gene expression. So that some of them could be very important. But as you know, roughly 300 million copies in the human genome of retroelements. So that probably most of them are not important for cells. But we don't know for the development. But this is one of the key, actually, concepts for my project is that by removing these important factors which could be important for the development in the first phase of the research, maybe we don't have to touch upon the ethics part, probably. And then by doing the mouse project, maybe we can see some elements which are important for development. But that could be different from the human process because retroelements are different between mouse and human. So yeah, so that's my idea.
So I have a kind of vague idea relating to this theme that you have exposed in thinking about interesting genome designs. I think something that could be interesting, probably a bit drastic for the moment, would be to segregate, let's say, arrange in some chromosomes very gene-dense kind of configuration. And in the context of neochromosomes, put all of these, for example, introns or retroelements or regulatory regions, and allow for example, by adding LOXP sites, allow for recombination to take place and just let this play out. And I would say that in the context of like an evolution experiment, I would say that if some regulatory or whatever transposon or introns are very important, they will be rearranged and co-opted back into the gene dense, let's say, chromosomes. And I think that could be quite interesting and a different way to try and understand which parts are very essential and which others are
Right.
maybe more context dependent.
Now I think
but
you.
it's very
Yeah.
drastic.
That's
and
all you're
like-
pursuing right now, right? So by doing this, so I think you are part of my project right now because yeah, that's what we are. There are many ideas and many hypotheses about the function of the retroelements, but I mean we want to stick to the function of the cells. So the function of the retroelements in the cell system.
Yes.
Yeah, by doing this, I mean maybe we can tackle that kind of questions, but I don't know. I don't have any idea yet.
Yeah, I think it would be very, very challenging, but it would just be very interesting
That's
to have this.
That's why I want to start with very small targeted gene regions,
Yes.
and then expanding to the whole genome scale. So it's time. So okay, if you're interested in, please grab me anytime during for the 2 days, and then discuss this theme. Thank you very much.